Hi, this video is to help you understand how to do today's activity. What we're going to do is we're going to use a restriction enzyme to cut segments of DNA at very specific places, and then we're going to count how many fragments there are and how many bases wide they are, and then you'll be able to plot it on a paper gel to figure out who did the murder. So here's how it works. So the restriction enzyme we're going to use today is going to cut at a very specific sequence, GAATTC. Has to be exactly GAATTC. Can't be GATC or GTCA. G has to be exactly GAATTC. And specifically, it's going to cut between the G and the A. So here we have a example piece of DNA. Remember, it's double-stranded and it's complementary, right? By that I mean C will pair with G, G will pair with C, A will pair with T, and T will pair with A. And that restriction enzyme, remember, is going to cut between the G and the A on GAATTC. So here's how you do it. You start going through the sequence on the top. Here we go. GAATTC. Here's our first one. So the restriction enzyme is going to cut between that G and that A. And remember, it's also going to cut between that G and that A in the opposite way on the bottom because it's the same sequence, only in reverse. Okay, we keep going down the sequence. Here we go. Here's another GAATTC. So it's going to cut between that G and that A, and then of course on the other side between that G and that A. And I hope that you see, instead of one piece of DNA, we now have three fragments. We have one fragment here, we have a second fragment here, and a third fragment here. So we now have three fragments, and we have to figure out how many bases wide each one is. Here's how you do it. You start counting from the end. In this case, you go from as far left as you can go to as far right as you can go on the fragment. So in this case, it doesn't matter where I start because they both start on the same level. 1, 2, 3, 4, 5, 6, 7. So this first fragment is 7 bases wide. We'll just mark it with a 7 so we can keep track. Okay. This second fragment here is a lot bigger. So we have to start here because that's right after the cut, and that's as far left as we can go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. But the fragment keeps going, so I have to keep going. 12, 13, 14, 15. So this fragment in the middle is 15 bases wide. Okay. Now let's do this last fragment. I have to start here right after the cut. 1, 2, 3, 4, 5, 6, 7, 8, 9. And again, they both end at the same spot. So this third fragment is 9 bases wide. So this restriction enzyme has cut at sequence GAATTC, and it's cut this piece of DNA into three fragments, one 7 bases wide, one 15 bases wide, and a third that's 9 bases wide. Now to plot this, you would come down to your gel. There's the sequences you're going to be doing. Now you're going to load in here, and remember, DNA always goes towards the positive pole because DNA is negative and opposites attract. So let's just use this one for an example, right? So the first fragment was 7 bases wide, so I go between the 6 and the 8, and I'll draw in a band. The second fragment was 15 bases wide, so I'll go between 14 and 16 and draw another band. And the last fragment was 9, so I'll go between 8 and 10 and draw a third band. Okay, you need to do that for all the sequences. Then you'll be able to compare the sequences and hopefully figure out who committed the murder.